Hi, yes. We're going to go over a brief overview of the nervous system, specifically the neural tissue or the nervous tissue. Okay, so we're going to look at that. So, unlike, a computer, portions of the brain, right? Because we talk about the brain as like a computer, but unlike a computer, portions of the brain can rework their electrical connections as new information arrives, right? And that's going to be part of our learning process. So we're going to talk about the nervous system, and along with the endocrine system, the nervous system also controls and adjusts the activities of other systems in the body, right? We're able to also provide a swift but brief response, and portions of the brain, again, can rework themselves. That's how we learn, right? So we're going to go over an overview of the nervous system, okay? The nervous system will consist of all the nervous tissue, okay? And it's going to include the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord, whereas the peripheral nervous system includes all of the neural tissue outside of the brain and the spinal cord. Okay, so as you can see here, the brain and the spinal cord in gray on the image, right? That's the central nervous system, and all the peripheries, right, will have their nervous tissue or neural tissue outside of that central nervous system. Okay. So when we're looking at the central nervous system, again, it's only associated with the brain and the spinal cord. It is responsible for integrating, processing, and coordinating sensory input, and is responsible for integrating, processing, and coordinating motor output, right? It's going to be the seat of intelligence, memory, learning, and emotion. The peripheral nervous system, again, is going to be associated with tissue outside of the central nervous system, right? Or neural tissue, right? Outside of the brain and the spinal cord. The peripheral nervous system will provide sensory information to the central nervous system and carry motor commands away from the central nervous system, okay? This peripheral nervous system can be divided into the afferent division, which brings sensory information to the central nervous system, and it begins at a receptor, right? So something, like a receptor in your skin. Okay, the efferent division, it will carry motor commands to muscles and glands, right? And it ends at an effector, right? So it ends like at a muscle, right? Or a gland or something like that. That's what we mean by effector. So the peripheral nervous system, again, can be subdivided into afferent, right? That can be further divided into somatic and visceral. And the efferent can be subdivided into somatic nervous system, SNS, and autonomic nervous system, right? So the autonomic nervous system can be further divided into parasympathetic division and sympathetic division. Okay. And here's a functional overview table going over that. Okay. How we have receptors such as special sensory receptors that will monitor taste, smell, vision, right? They'll be able to using the peripheral nervous system, that neural tissue that is able to connect to the eyes, right? Connect to the olfactory bulb, right? Connect to the tongue. They'll be able to take information, sensory information, to the central nervous system, so to bring that information back up to the brain and the spinal cord, right? So this is just a great image right here showing the major anatomical and functional divisions, like the central nervous system and the peripheral nervous system. It also goes over the autonomic nervous system in that top left box right there, where the autonomic nervous system is components of the central nervous system and peripheral nervous system that controls our visceral functions, right, our visceral organs. Okay. We're also going to look at the gross anatomy, specifically of the spinal cord. We're going to look at the histology of the neuron. Okay. So a neuron, right? So nervous tissue consists of two types of cells, right? You have neurons. These are nerve cells that are responsible for the transfer and processing of information in the nervous system. And then you have its bodyguard, right? The neuroglia. These are those supporting cells. These will protect the neuron, okay? These will protect the neuron. So let's look at the neuroglia, right? Let's look at these protectors first. We have astrocytes, right? And so this is going to be neuroglia, right? Or these glial cells, these supporting cells of the central nervous system. There's going to be 4 of them, okay? We're going to have astrocytes, we're going to have oligodendrocytes, we're going to have microglia, and ependymal cells. Let's first start off with astrocytes, okay? So astrocytes, these are going to be the largest and most numerous glial cells, okay? They have a variety of function, however, they're also poorly understood, right? But they make up the blood-brain barrier, okay? They provide structural support. They regulate ion, nutrient, dissolved gas concentrations. They also absorb and recycle neurotransmitters, and they form scar tissue after injury, okay? Another neuroglia or glial cell of the central nervous system is the oligodendrocytes, right? The oligodendrocytes, they form that myelin sheath, right? Or those internodes, right, of myelin sheath, okay? These regions that are dominated by the myelinated axons, right? So they form these myelin sheaths over the axons, okay? And they also provide structural framework, okay? These regions that are dominated by the myelinated axons, they constitute the white matter, okay, of the central nervous system. The areas that don't have myelination, right, that are devoid of the myelinated axons, that's the gray matter, right? The gray matter is going to be composed of the cell bodies and the dendrocyte, or the dendrites, sorry, okay? So again, white matter is going to be the myelinated axons, right? The gray matter is going to be the cell bodies and the dendrites. Now the third neuroglia cell of the central nervous system is going to be microglia, right? Microglia, okay? These are going to be the smallest. They possess, a slender cytoplasmic process with many fine branches, okay? You can see the microglia in the middle. So these are phagocytic cells. Okay, these are phagocytic cells of the central nervous system, engulfing cellular debris and waste products, right? So they remove that cellular debris, they remove waste and pathogens by phagocytosis, right? Engulfing them. Our last neuroglia of the central nervous system is the ependymal cells, right? So the ventricles of the brain and the central canal of the spinal cord, they're going to be lined by the cellular layer called the ependyma. Okay, so these ependymal cells, right, they're going to be cuboidal and columnar in form. They're going to be involved in the cerebral spinal fluid production, right? And again, you can see on the image in the pink, right, they're going to make up that cellular lining called that ependyma. Okay, so they line the ventricles of the brain and the central canal of the spinal cord. Right? Again, they assist in producing, circulating, and monitoring that cerebral spinal fluid. Okay, so those were the 4 neuroglia or glial cells of the central nervous system. Okay, we also have 2 neuroglia or supporting, glial cells of the peripheral nervous system. Right, so the cell bodies, right, when you have just cell bodies in the peripheral nervous system, when they're clustered together, that's called ganglia. Okay, the axons that are bundled together in this peripheral nervous system, they're called nerves or peripheral nerves. Okay, there's going to be 2 types of neuroglia cells in the peripheral nervous system. You have satellite cells, they're going to surround the cell bodies. You have Schwann cells, they're going to surround the axons. Okay, and you can see in the image right here, the satellite cells, they just simply surround. They will surround that cell body, right, protecting that cell body. Whereas the Schwann cell is going to surround the axon, similar to the oligodendrocyte, how they form that myelin sheath in the central nervous system. The Schwann cells form that myelinated, sheath over these axons in the peripheral nervous system. Okay. And this just shows you an image of a myelinated axon versus an unmyelinated axon in the peripheral nervous system. You see how that axon's in the middle, and then that myelin sheath or that Schwann cell will rotate, right? And the inner membranous layers are compressed and the cytoplasm is forced into more superficial layers. And when it's completed, the myelin sheath consists only of the phospholipid bilayer of the plasma membrane with the Schwann cell nucleus and cytoplasm at the surface. Okay. So let's look at a neuron, right? The main cells, well, one of the type of cells, right, of the nervous system. Okay. So just looking at the cell body, right? The cell body of a representative neuron will contain a relatively large round nucleus with that prominent nucleolus in the middle. Okay. The surrounding cytoplasm constitute the perikaryon. Okay. That's going to be the cytoplasm, like perikaryon, right? And the cytoskeleton of the perikaryon contains neurofilaments and neurotubules. And we have bundles of neurofilaments called neurofibrils. These are going to be those cytoskeleton elements that extend into the dendrites and the axon, okay? Help with the structure of the cell. So this perikaryon, it contains organelles that provide energy and perform biosynthetic activities, right? The numerous amount of mitochondria that we see free and fixed ribosomes, we see the membranes of the rough endoplasmic reticulum. They're going to give this perikaryon a coarse, grainy appearance. Okay, so the mitochondria is going to generate ATP to meet the high energy demands of an active neuron. The ribosomes in the rough ER, right, that's going to synthesize peptides and proteins. You're going to have groups of fixed and free ribosomes, right? They're going to be present in large numbers. And these ribosomal clusters, they're called chromophilic substances or Nissl bodies, as you can see right there in the bottom left. These chromophilic substances or Nissl bodies, right, they account for the gray color of areas that contain neuron cell bodies, right? So when we say like it's gray matter, right, seen in like a gross dissection of the brain or the spinal cord, is because of those clusters of the free ribosomes, those Nissl bodies, right? So most neurons, they lack a centrosome, right, which we'll get into in just a second, right? And in other cells, the centrioles of the centrosome, they form the spindle fibers that move chromosomes during cell division. So neurons usually lose their centrioles during differentiation, and they become incapable of undergoing cell division. Okay. And so you can see these are the different parts of the neuron. We also have the dendrites, right, on the end, those finger-like extensions, right? They're going to be stimulated by the environmental changes or the activities of other cells. We have that axon. That's going to be able to conduct a nerve impulse or that action potential, right, towards the axon terminals, like the end of the axon. You can see a little piece coming off of that axon right there. We also have the axon terminals, of course, right? The axon terminals, they will affect another neuron, okay? Or, you know, another muscle or gland, right? Some type of effector organ, right? We also have the axon hillox. That's the area where the initial segment of the hillox will connect to the cell. The initial segment of the axon will connect to the cell body, right? We have the axoplasm. That's just the cytoplasm of the axon. That collateral, that was that little branch that came off of the axon, right? And the ends of the axon and the collaterals, that's going to call it the telodendria. And of course, the axon terminals is the end of the telodendrias or the end of the axon, right? And axoplasmic transport, that's just the movement of materials between the cell body and the axons. Okay. Here's another representative axon, right? Representative neuron right here. You can see the cell body up top, right? Having that initial segment of the axon, which will, you know, be the beginning of that axon. And as we move down the axon, that's where that action potential will move. Okay. So neurons can be classified based on structure or function. The structural classification, right, is based on the number of processes, right, extending from the cell body. Whereas the functional classification, it's going to be either sensory, motor, or interneuron. And those interneurons are going to be involved with both sensory and motor neurons. And you'll see how those interneurons are basically going to be between the sensory and motor neurons. They're literally going to be situated between sensory and motor neurons. Okay? So the structural classification of neurons, right? We have an axonomic neuron, right? It has many processes, but we can't differentiate between the axons and the dendrites, and they're only found in the central nervous system. Okay, that's A on the left. We have a bipolar neuron, the second one, right? That's where the cell body is between the dendrite and the axon, and the axons are not myelinated in this one. We have the pseudo unipolar neuron, right? And this is where the cell body is just off to one side of the axon. We have a multipolar neuron, right? This is going to be the most common type of neuron in the central nervous system. And it's typically going to have a single axon and multiple dendrites, okay, as you can see right there. That's that most common neuron of the central nervous system. And then they can also be classified based on their functions, right? So there's three functional groups of neurons: sensory neurons, motor neurons, and interneurons. Sensory neurons, right? So they form the afferent division of the peripheral nervous system, and their functions is to deliver information to the central nervous system. Okay. Again, their axons are afferent fibers, and they send information from the peripheral nervous system to the central nervous system, right? So there's the afferent division, those afferent fibers are those sensory neurons, okay? And there's going to be somatic sensory neurons and visceral sensory neurons, okay? There are three categories of receptors. You have interoreceptors, which they monitor the internal organ activity. You have exteroreceptors. They provide information about the external environment. And you have proprioreceptors, or proprioceptors, where they monitor your position and movement. Okay. You have motor neurons, right? The efferent division of the nervous system is going to be composed of motor neurons, right? So their axons, these axons of these motor neurons, these are going to be efferent fibers, right? Their axons are going to be the efferent division, okay, of the nervous system. So they're going to be able to send information from the central nervous system to the peripheries, right? So from the central nervous system to the peripheral nervous system is going to consist of two divisions, right? The somatic nervous system, which will consist of somatic motor neurons, and you have the autonomic nervous system, and that's going to consist of visceral motor neurons, okay? There's also the autonomic nervous system is also going to consist of preganglionic fibers and postganglionic fibers. So the axons extending from the central nervous system to a ganglion are called preganglionic fibers, whereas the axons connecting from that ganglion to the rest of the, the peripheral effectors, they're going to be known as the postganglionic fibers. Okay. And as you can see right here at the bottom, right, it goes from the central nervous system, right? Those motor neurons, those visceral motor neurons are able to send those efferent fibers, right? And the preganglionic fibers, right? They're right before they get to the ganglion, and right after they meet that motor ganglion or ganglia, they become postganglionic fibers, okay? And then they are able to synapse with an effector, such as the smooth muscle glands, cardiac muscle, or adipose tissue. So the other functional class of neurons are those interneurons, okay? Those interneurons are located entirely in the central nervous system, okay? Entirely in the central nervous system. They are going to be situated between the motor and the sensory neurons, okay? They analyze sensory input and they coordinate motor outputs, right? They can excite or they can inhibit neurons, okay? And so this is what I mentioned earlier, where most neurons, they lack a centrosome. And then remember the centrioles of the centrosome, they form the spindle fibers that move chromosomes during cell division. And so if they don't have that, the neurons usually lose their centrioles during differentiation. And thus they become incapable of undergoing cellular division. Right? So if these specialized neurons are later lost to injury or disease, they can't be replaced. Now if there's an injury, right, to like a peripheral nerve, right? So that we discussed the steps involved in Wallerian degeneration and nerve regeneration, right? So neurons, they have a very limited ability to recover after an injury. Okay, very limited ability to recover after injury. So following an injury, the chromatophilic substances, they'll disappear and the nucleus moves away from the centralized location within the cell body. And if the neuron regains normal function, it will gradually return to a normal appearance. So if the oxygen or nutrient supply is restricted, such as like in a stroke, or mechanical pressure is applied to the neuron, as often happens in like those spinal cord or peripheral nerve injuries, the neuron may be unable to recover unless circulation is restored or the pressure is removed within a period of minutes to hours, right? But of course, if these stresses continue, the affected neuron will be permanently damaged or killed. So in the peripheral nervous system, Schwann cells participate in the repair of damaged nerves, right? And that's this process called Wallerian degeneration, right? And so the axon distal to the injury site deteriorates and macrophages migrate in to phagocytize the debris. Okay. The Schwann cells in the area, they're going to divide and form a solid cellular core that follows the path of the original axon. And additionally, these Schwann cells, they release growth factors to promote the axon regrowth. Okay. And so if the axon has been cut, right, new axons may begin to emerge from that proximal end of the cut within a few hours. However, in more common crushing or tearing injuries, the proximal end of the damaged axon will die and regress for 1 centimeter or more, and the sprouting of a new axonal segment may be delayed for 1 or more weeks. But as the neuron continues to recover, its axons grow into the injury site, and the Schwann cells wrap around it, right? The growing axon is most likely, to arrive at its appropriate destination if the damaged proximal and distal stumps remain in contact after the injury. Okay, so that's an important one. There is limited regeneration that can occur inside the central nervous system, but that situation is a little bit more complicated because, number one, many more axons are likely to be involved. Number two, astrocytes, they produce scar tissue that can prevent axon growth across the damaged area. And also astrocytes, they release chemicals that block the regrowth of axons, right? So that's why you don't really get a lot of regeneration or in the central nervous system, okay? So what about a nerve impulse, right? What's a nerve impulse? So the ability to conduct the impulse is going to be known as excitability, okay? And that's the ability of the plasma lemma, or the plasma membrane, to respond to an adequate stimulus, okay? So a nerve impulse is known as the action potential, right? The action potential of a nerve is the change in membrane potential that develops after the plasma lemma is stimulated to a level known as the threshold. Right? We have membrane potential. Membrane potential is the uneven distribution of positive and negative ions across that plasma membrane. And so a stimulus creates a change in membrane permeability. And so if it meets that threshold stimulus, right, and that's a level of stimuli to cause a change in membrane permeability, that impulse will propagate along the axon. Okay, so this is how we can create a nerve impulse, okay, by altering or changing that membrane potential. Okay. And so a myelinated axon can conduct impulses 5 to 7 times faster than an unmyelinated axon, right? So the speed of an impulse will depend on the presence of a myelin sheath, right? So if you have that myelin sheath, you're going to have a fast impulse. You lack or you don't have that myelin sheath, then you're going to have a slow impulse, right? The myelinated axons with a large diameter, they have a fast impulse of up to 140 meters per second, whereas those unmyelinated axons with a smaller diameter, they have a slow impulse, right? Relatively less than 1 meter per second. So synaptic communication, right? So a synapse, right, is the junction between a neuron and another cell. It could be another neuron, or it could be a gland, it could be a muscle cell, right? There's going to be 2 types of synapses. There's chemical synapses. These are called vesicular synapses. And we have electrical synapses. These are called non-vesicular synapses, okay? So these are just some of the structures of a synapse, right? Number one shows a synapses with another neuron. Okay, number two shows that neuromuscular junction. Okay, a synapse from the neuron with a muscle cell. And we have a neuroglandular junction, right? The synapse between a neuron and a gland cell. Okay. So chemical synapses, or these vesicular synapses. So vesicular synapses, right? They are by far the most abundant type of synapse, right? And it involves the passage of a neurotransmitter substance between cells. So most interactions between neurons and all communication between neurons and peripheral effectors will involve a vesicular synapse, right? So however, a neuron communicating to another neuron, right, is going to be a vesicular synapse. It's going to be this chemical synapse. Or if it's the neuron communicating to that peripheral effector, right, it's going to be a vesicular synapse. Okay. So at a vesicular synapse between neurons, a neurotransmitter released at that presynaptic membrane, right, of the first neuron, of that terminal end, will bind to a receptor protein on the postsynaptic membrane, right, so neuron number 2, okay, and it's going to trigger that transient change in the transmembrane potential of that receptive cell, and only the presynaptic membrane releases a neurotransmitter, right, so as a result, communication occurs in one direction only, and from the presynaptic neuron to the postsynaptic neuron, right? So again, communication is only going to occur in one direction, okay? You have electrical synapses or non-vesicular synapses, right? These is where impulses can be conveyed in either direction, right? Or both directions, really. And so these are going to be found between neurons in both the central nervous system and the peripheral nervous system, but they're going to be relatively rare though, right? So at these synapses, at these non-vesicular synapses, the presynaptic and the postsynaptic membranes are tightly bound together, and communicating junctions permit the passage of these ions between the cells, okay? And because these two cells are linked in this way, they function as if they shared a common membrane, and the nerve impulse crosses from one neuron to the next without delay. So in contrast to the vesicular synapses, these non-vesicular synapses, they can convey nerve impulses, again, in either direction. Okay? So looking at the neuron organization and processing, neurons can be organized into smaller organized groups, right, called neuronal pools, okay? The neuronal pools are identified by their neural circuits, right, such as divergence, convergence, serial processing, parallel processing, and reverberation. Divergence, right, is the spread of information from one neuron to several neurons. It permits broad distribution of specific input, right? And the information enters the central nervous system and then spreads to the brain and the spinal cord at the same time. So you can see it's coming from one neuron to several neurons, right? That's divergence. It's diverging into multiple neurons. Now the opposite, we have convergence, right? That's where a circuit is providing input to a single neuron from multiple sources, right? Again, the information going from several neurons to a single neuron, right? So movements of the diaphragm muscle are involuntary, right? But yet, at times, we can also move the diaphragm muscle voluntarily, right? Especially when we take in a deep breath, right? Serial processing, okay? That's information going from one neuron to the next in a sequence, right? So that's this circuitry is sequential, right? This is where, an example, information going to one part of the brain, then it goes to another part, and then it goes to another part. But it has to go through part 1 first to get to part 2 to get to part 3. Okay. And then we have parallel processing, right? This is where several neurons are processing the information at the same time. So for example, if you step on a nail, you typically move your foot, you might shout ouch and dance a little bit, right? Moving your foot, saying, oh, ah, all at the same time, right? So this is basically a circuit in which the neurons or pools process the same information simultaneously. Then we have reverberation, right? This positive feedback circuit. So those collateral axons, right, those axons that we're extending off, right, those collateral axons, they extend back toward the origin of the impulse to cause an enhancement or a continuation of that impulse. Okay. And again, these are, how, the billions of interneurons within the central nervous system are organized into a much smaller number of these neuronal pools, right? Divergence, convergence, serial processing, parallel processing, and reverberation. So when we're looking at neurons in the peripheral nervous system, right, versus those neurons in the central nervous system, neurons in the peripheral nervous system, they consist of ganglia, right? So their cell bodies are going to be termed ganglia, and the axons are bundled together in what we term nerves or peripheral nerves. And the central nervous system, the collection of these cell bodies, they're called a center or a nucleus or nuclei, right? Where the white matter consists of axons and bundles called tracts, okay, or columns, okay? Axons in the central nervous system are called tracts or columns, and the cell bodies in the central nervous system are called nuclei or centers, whereas the neurons in the peripheral nervous system, the axons are bundled together in nerves, and the central or the cell bodies are the collection of those cell bodies in the peripheral nervous system are called ganglia. Okay, and this just goes over the again the gray matter is going to be the ganglia. Right, ganglia is a collection of neurons, cell bodies in the peripheral nervous system, and the white matter in the peripheral nervous system, right, that's going to be the nerves or the bundle of axons in the peripheral nervous system. Okay. When we're looking at the central nervous system, the gray matter organization, we have it being the neural cortex, right? That's the gray matter on the surface of the brain. And again, the nuclei, which is a collection of neuron cell bodies in the central nervous system. And then we have the white matter. That's going to be those axons, right, of those neurons that's in the central nervous system. Okay, and that was just a brief overview of the neural tissue, right, in the central nervous system. Thank you.